right. I'll look at what you said here.
So it's under here.
All right.
That's it.
All right. All right.
And advance.
Yes. If you want to, yeah, if you want to use, you can use a keyboard, or if you want to use, we have a handheld. If you want to use, obviously that one's forward.
Are you beaming this somewhere?
Yes, we teleconference to our VA facility.
Okay, because usually they like you to use this arrow.
Yeah, they'll be okay. It's actually a high def feed, so you're.
The VAs, there's 6 other places that you can use.
Yeah. The red button is a pointer.
Okay.
We'd like to use that. I'll adjust the lights. They won't be shining like that in a minute. But I'm going to put this on you. It's turned off. When you're ready, Dr. Roman will give an introduction. When you're ready to start speaking, you come up to start, just hit this button on top so the light turns green. Turn this off. I have a reminder here. It's a reminder to turn on the microphone as well. We had someone that didn't once, so.
Okay, we're ready?
Have at it.
Oh, I can't read without the light. Okay, no, I'm kidding. Good morning. Welcome to the Department of Medicine Grand Rounds. Every year we come together to celebrate the life of Nancy Middleton Smith. Lucille Gibson and Dr. Smith Gibson, who was a grandson of Nancy Smith, established this lectureship in honor of their grandmother, who was born in Everett, Kentucky in 1874. She was thought to be a very intelligent woman who didn't have formal schooling on herself, but had encouraged her children and grandchildren to become educated to their fullest capacity. And she was extremely proud to know that her grandson and 2 great-grandsons graduated with honors from the School of Medicine at the University of Louisville, and then a granddaughter from the School of Law at this same institution. He devoted her life to feeding the poor and ministering to the sick, but she suffered periodically from cardiopulmonary disease, and in fact died of pulmonary edema in 1927 at age 53. And because of this reason, Dr. Gibson and his family has set this lectureship, and every year we bring a speaker that talks either about cardiology or about pulmonary medicine, and this year is going to be about pulmonary medicine and a bit more, as you will see in a minute. The Gibsons has also provided resources for a chair endowment that is currently held by Dr. Aruni Bhatnagar, who was with us last night, and we had a wonderful dinner with our guest speaker. Before I bring the speaker, a little bit about history. Think about when this lady was born in 1874. If you look at the British Medical Journal archives, there were articles about syphilis and the smallpox epidemic about 1870 and 1874. If you look at the New England Journal of Medicine in March of 1874, they have articles about periostitis, paremia after wound infection. They talk about Dr. Boucher-Fontaine. I'm going to say that again. Boucher-Fontaine, who was doing research about how quinine had antiseptic activity. But there were interesting 2 events that happened in 1874. One of them, you'll find interesting, was an article published at the American Medical Weekly, which was at the the Louisville Medical Journal. Was published in 1874 by Legrand G. Capers. It's called Attention Gynecologist. Have you heard about this? So he was an Army doctor. In fact, he was a field surgeon that worked with the Confederate Army. And in Mississippi, he described the case of a young soldier who was hit by a bullet in a testicle. The bullet traveled into a house and hit a woman in the abdomen, and months later, she delivered a baby. Now you are laughing, but that story was considered true and in fact was republished later on until 1959 as the latest time it was published as a true event. It turns out it was not true. He wanted to poke fun at all these stories about the Civil War that was happening. Another event that happened in 1874 was the presidential address by John Tyndall. He was professor of natural philosophy at the Royal Institute. And he gave the presidential address of the British Association of the Advancement of Science. And he started talking about ancient Greece. He started talking about Middle Ages, how then Galileo and Copernicus changed and revolutionized medicine. But at the end of his conversation, he began to separate and dissect the distinction between religion and science. And this created tremendous controversy that affected his career for the rest of his life. He said, religion, this is 1874, religion adds inward completeness and dignity to man, but it is restricted to the region of poetry and emotion. The region of objective knowledge belongs to science alone. Any systems that infringe upon the domain of science must submit to its control. Now think about that statement back in the early or late 1800s. In fact, Protestants and Catholics just revolted around this, and scholars till today consider that an important event in the continuing war between science and religion that we still see today. So I told you about these events that are happening and how infection was so important. Little did we know at that time that we would think of bugs in a very different way today. And that's what this lecture is all about. So let me introduce you about our special guest, Dr. Homer Twigg. Dr. Homer Twigg was born in Washington, D.C. In 1983, he obtained his MD from Georgetown University. He was an intern and resident in internal medicine at University of Alabama in Birmingham. And then in 1989 was recruited as assistant professor at Indiana University, where he has remained ever since. And I met him about a year later. We were just discussing that last night. He's been the recipient of the Edward Livingston Trudeau Award from the American Lung Association. He also received the Murray Auerbach Award, which is the highest volunteer award given from the American Lung Association of Indiana for his efforts for patients and so forth. He's an outstanding clinician. He's an educator and a mentor. He had served as a program fellowship director at his institution, but has been division director for pulmonary and critical care since 2002 and remains to do so. But he's also a scientist, and he's written extensively. He's been one of the few, I think, pulmonary doctors who've been engaged actively in research related to lung and HIV infections since the early 1990s. He has served on important NIH study sections. He's currently on one of the lung NIH study sections. He's reviews for prestigious journals. And he is funded currently to study the lung microbiome. And I think you're going to be excited today to hear that bugs are not just bugs, bugs are us. So I leave you with Dr. Homer Twigg.
Okay, thank you, Jesse, for that introduction. I also am grateful for the invitation to present at this named lecture. I hope I do it honor. So today I'm going to talk about the respiratory microbiome, and my goal today is to basically provide you a primer, sort of microbiome 101, on why this is important. This is sort of an emergent area of study. And I want to introduce the concept of a microbiome. I'm going to talk obviously a lot about the lung, but actually most of what we know about microbiome is in other organs. We're going to describe how the microbiome is measured. We're going to try to give you some idea, because when you read the literature, it can be daunting. So I'm going to try to give you some terminology and introduce to you the background of how a microbiome is analyzed. I'm going to talk to you about how the environment affects the microbiome and how the microbiome is important in health and disease. But since this is a grand rounds, I was told I had to start with a case. So I have two cases. The first is a case of Heinz and Obi, who were two baby mice. They were born after an uncomplicated pregnancy, have a healthy childhood, eat the same food, and they're very active. They race around in their cages. They love running around, seeing who's the fastest. However, Obi seems to be slowing down some. He can't keep up like he used to, and he gets tired easily. He's on no medications. These mice are not destined to some sort of study protocol. He's a non-smoker. Similar illnesses occurred in obese parents. No shortness of breath, no fever, chills. And on physical exam, this is Heinz. He looks like a typical mouse. And this is Obi. And it's easy to see why Obi is no longer able to keep up. And so the diagnosis for Obi is obesity and deconditioning. The second case is Butch and Dexter. And these are twins born after uncomplicated pregnancy, normal first 2 years of life. They eat the same food, both type and amount. And around the age of 3, Dexter begins having episodes of cough, shortness of breath, getting antibiotics all the time, missing in more and more school. He's on no regular medications, but is on frequent antibiotics. They have similar travel history. Family history is unremarkable, and review systems is negative. The social history reveals that Butch likes to play outside, and he loves his dog, and so he's always crawling around on the dog, whereas Dexter likes to watch TV, and when he got old enough, spent a lot of time in front of his computer. And on physical exam, Dexter has the runny nose, the shiny eyes, he's got a few wheezes, and his diagnosis is allergies and asthma. And so what I'm going to try to talk to you today is about how the environment and the microbiome might define some of these clinical characteristics that we see. So what exactly is a microbiome? Anytime you want to create something of importance, just stick an "ome" at the end of it. So you have a genome, and if you put an "ome" at the end of it, it's going to be important. So you can get a proteome, you can get a lipidome, and then most recently, you can get this microbiome. And why is this important? It's important because the NIH gives you money to study these things. The Genome Project was a huge undertaking, and now the Microbiome Project is a huge undertaking, and is now part of the NIH roadmap. And it's estimated that microbes outnumber human cells by 10 to 1, and this is bacteria. If you look at viruses, it might even be 100 to 1,000,000 to 1, outnumbering the human cells. I'm going to only briefly mention viruses at the end. Most of this talk is going to be centered on bacteria. And I changed this slide a couple days ago. When I first wrote it, it was 99% of bacteria can't be cultured, but with new culture techniques, we're probably up to 5 to 10% we can culture, and 90 to 95% of the bacteria that are out there that we are colonized with cannot be cultured. And this is getting a lot of lay press. This was Science News a few years ago talking about the 600 bacteria that colonize our mouth. And I'll have one slide about how these mouth bacteria may be important in defining certain phenotypes. It's important to remember when you look at the microbiome that there's 3 possibilities these bacteria can share with us. They can be commensals, which means they just live with us and they don't do anything. They can be potential pathogens, and these are your pathobionts. And these, I'm going to argue, in some way, not only, if there are too many, they can be pathologic, but they can sort of define our immunologic background, our sort of inflammatory background against which we encounter antigens. And then you have the things that are good for us, the symbionts. And these are things that define how we digest food, for example, what our susceptibility to cancer might be. And I'll give you examples of this. You get immunologic disequilibrium when the pathogens seem to increase. And so this is what causes disease. And what we strive for is to be in this balance with the organisms we are colonized with. To understand how we do the taxonomy of a microbiome, you need to understand the tree of life. If you believe in Aristotle, that's kind of interesting. You got the historical background from Jesse. If you believe in Aristotle, everything begins at the bottom with life. You're either alive or dead. But the family tree actually begins in domains. And when you create a microbiome and describe the bacteria, you basically are going to do sequencing, which I'm going to show you how it's done, up to the genus level. And you can see here the humans, in honor of the Oscars, and E. coli. And you can see where both bacteria, eukarya, and so on as you go up the family tree. And viral taxonomy begins at the order. And what's important here is you can see animals and fungi. We are very close to fungi, but in this phylogenetic tree, we're pretty far away from bacteria. And this is the basis for the sequencing that leads to microbiome analysis. So how do you measure a microbiome? And it's all based upon sequencing a ribosome. Ribosomes are essential for processing amino acids and proteins. And because they're essential for creating these proteins, they are among the most conserved genes that we have. And if you look at humans, we have 18S small subunits of our ribosomal RNA. And bacteria and fungi, for that matter, fungi are 18S. Bacteria are 16S. So everything we do is based upon sequencing a 16S gene. The 16S ribosomal RNA is present in all bacteria, and it's about 1,500 base pairs, and they're these conserved regions, as you can see in the red. And you can put primers into these conserved regions and sequence this whole gene. Now, my concept of what happened when I first got into this is that you had this sample that you want to test it, and you gave it to somebody who put it into this black box, and for $100 you would get all your sequences that you wanted. But in actuality, it's a little more complicated than that. And this is basically how they sequence. And I'm going to do very brief, trying to give you an introduction of how this sequencing is done so you can hopefully understand this process. And what you have are primers, and you can either sequence the whole gene or you can sequence just a part of it. And most people, and what we are doing is just sequencing what's called the V1 through V3 region. So you have a primer. What you do is you amplify the DNA, and then you put your amplified DNA in a test tube with a bunch of bases and some bases that are labeled, and these labeled bases are terminators. They end the sequence if that base happens to get attached to your DNA that you've got. So, for example, you have this DNA, and the first thing that will bind to this DNA will be an A, and it can either be a normal A, or it can be one that's a chain terminator, and it has a dye to it. So if the A stops, that means there's no more additional bases that are added to the end of this, and at the end, when you pull these out by size, you'll have this tiny little fragment that has a blue tag on it. Let's say the A is not hit by a dye, and the second one is. The C comes in, and it has a terminator in it. It's tagged red. That means you'll have a piece at the end that's a little bit bigger, and it ends in a C. It ends in a red dye. And if you do this, you'll be able to sort of slowly build these things up. At its extreme, you get the whole gene sequenced, and the last thing that's added is a colored G. And after this, the sequencing stops. So this is the basis of this sequencing. And once you do this, you have these various sizes, as I showed, and you just basically put them together, and you have your sequence. And then you go to a database, and you're able to identify the bacteria. Now, if you read this slide, your eyesight is better than me, but this is the type of data that you get when you put this in. And this is one sample, and this one sample has all these different bacteria into it. And when you look down at one of those lines or two of those lines, this is what you see. You have the bacteria, the Proteobacter, down to your genus level. And these numbers are how confident you are that this is real, based upon the database that's out there. We are very dependent on the database. If the database is inaccurate, then your identification of bacteria is inaccurate. So you can see that, at least in this one sample, at an RDP classifier, that means they want 80% match of the sequence that you've done with what's in the database. We are 100% confident that this is Ralstonia. We're 100% confident that this is bacteria. There's some bacteria, for example, where you're sure they're bacteria, but when you get down to the genus level, you're only 51% sure because the coverage is not as good. And so you're very dependent on your databases. So what is this used for? What has this sequencing been used for in the past? It's used for accurate identification of bacterial species. It's been used to define virulent versus avirulent strains. It was used to identify rapid identification of mycobacteria. This can be done to quickly figure out if you have mycobacteria in your specimen. You can find new species, and you get these bacteria that are not easily cultured, either because you're on antibiotics or because they're just not culturable with today's technology. And in fact, Bartonella, bacillary angiomatosis, and Whipple's disease, this is how these organisms were discovered through this type of sequencing. There are problems with this. And so when you sequence this 16S gene, you're basically looking at a species level. And there is a disconnect between this sequence, this gene, and phenotype. And that's the problem you have when you start trying to put genomics with phenotype, taxonomy. So we have identical 16S ribosomes to this guy. But I would argue that most of us don't look like this guy. But according to 16S sequencing, we're identical. We also are fairly close to this guy, 98.6%. So the question is, how much different do you have to be considered a new species? And that's one of the problems that this field grapples with. And there are lots of examples of this sort of disconnect. For example, you can have these two Clostridia, which are very much different in their 16S RNA, and yet they're both Clostridia. And that's considered a problem. On the other hand, you have these organisms that have a similar phenotype, Mycobacterium tuberculosis, Mycobacterium bovis, who have identical 16S sequences, and yet we know they have completely different types of diseases. So you have to keep that in mind when you're identifying these organisms. And in fact, this is just I consider this a fingerprint. When you do this on a sample, you're just identifying the bacteria that are present. Does not imply any sort of cause or effect of the phenotype. So to try to get around this, a new field was generated. And if you put the meta in front of something, that means it's more comprehensive, it's transcending. And so you can have metaphysics, you can have meta-analysis, you can have meta-browsers, you can have meta-programming, or you can have metagenomics. And metagenomics is an attempt to not only get the bacteria sequenced, but all the bacterial genes sequenced and all the host genes at the same time to see if the bacteria that you are identifying are having any sort of impact on the host. Genomics is the determination of complete genetic complement of an organism, and this is the basis of the Human Genome Project. Metagenomics not only looks at that material, but it looks at the bacteria that are from the same environment. And it attempts to try to relate the bacteria that we identify and the genes the bacteria express with the host. Again, my concept of this is you took a sample, this time for $3,000, you put it into a black box, and you got an answer. And this is the only complicated slide I'm going to show on this, because this is shotgun sequencing. And in contrast to 16S, where you're really targeting a specific gene, in shotgun, you sequence everything. So you just break up a gene, and you have all these different fragments, and what you're trying to do is to find these fragments that overlap. And if you find fragments that overlap like this, you basically know that somehow these are connected. And so this one, for example, is connected to this, and you sort of build this whole gene. This is called coverage, and the more like areas that you have, the better your coverage is, the more confident you are that this gene is actually present. All right, so that's how you determine a microbiome. That's how you look at it. But how do you analyze it? In order to understand the microbiome, you have to understand the terminology. And we talk about things called diversity. There are two types of diversity. There's alpha and beta diversity. So imagine you have two populations, A and B here. And you have 10 species. And in population A, you have 10 of each. And in population B, you have 1 of each, except for the last one, you have 91 of species J. These two populations have the same richness. They each have 10 different bacteria in them. Population A is more even, and therefore this is more diverse than this population, which is dominated by a single bacteria. And the indices that are frequently used to define alpha diversity are Shannon and Simpson indices. Beta diversity compares between two populations. Alpha diversity is the diversity within a single person. Beta diversity is the diversity of this audience. So in population A, you have 10 different species. Population B, you have 9 different species. But A has a 1 and 2, which is not in B, and B has a number 11, which is not in A. So the beta diversity of this population is 3, because those are the distinct species in these populations. This has an alpha diversity of 10. It has 10 different species. This one has an alpha diversity of 9, so they're pretty similar from an alpha diversity. But the beta diversity, the differences in this population is 3. And you can sort of look at it in a dendrogram. This is sort of how it might be presented, where the more overlap you have, the closer you are on this endogram. So June and Sam, here's June and here's Sam. They have a lot of overlapping organisms, where John is significantly different from June, and you can see John is the furthest away from June on this type of dendrogram. Now, when you look at these types of papers, typically this is what you get. You get these sort of mind-numbing sort of presentations that come up, and you look at these and your eyes begin to glaze over, and you're trying to figure out what are these things. I mean, this is sort of difficult to understand. Well, what these are in its simplest form are heat maps. And so each one of these for persons on this dendrogram is a person, and these are all the bacteria that are lined up. And the darker your bar, the more bacteria there are in a certain place. And I'm going to come back to this slide near the end as I talk about some data. The other way you show beta diversity is through this principal coordinate analysis. And essentially, when you look at this, these two people right here, for example, have virtually the same bacteria. They're almost identical, whereas this person and this person are completely different. So it defines how close the population is. And you could argue that this orange population is pretty closely tight. That means most people in this orange population have the same type of bacteria, whereas all the blues, they're kind of scattered. That means there's a diverse beta diversity here. There's a lot of different people. Now back to this slide, when the NIH roadmap first came out, they looked at nasal, oral, skin, and GI, and obviously what was missing from my perspective was the lung. And there really is a true debate out there about whether the microbiome exists in the lung. The lung has always been thought of as sterile. And as you know, to get to the lung with a bronchoscope, you have to go the veritable sewer that is our mouth. And so there's this great debate of whether there really is a respiratory microbiome. And I've always argued you need to find the signal in the noise. I believe that there is a respiratory microbiome. And this debate sort of stemmed from this very first paper that came out from the Penn group. Charleston, Ron Coleman's the senior author on this. And they did a very careful study where they looked at BALs and all sorts of samples as well as environmental controls. And basically what they found is that in BALs, the things that were different, and what you're looking for on this heat map are dots. So what's unique about all these lung specimens? Well, here's the BAL. Well, what these BALs were, at least in their study, was all environmental contaminants. That means they just took stuff, ran it through their scope, sequenced it, and anything they found that was unique in BAL actually was in their environment at Penn. I'm not going to, I have a whole talk defending the respiratory microbiome, but I'm not going to, that's not the purpose of this talk. But I believe there's real evidence for a respiratory microbiome, and the LHMP, the Lung Human Microbiome Project Group, wrote a paper that was in Lancet Respiratory Medicine, basically outlining these reasons why we think one really does exist. And in a nutshell, there are differences between mouth and lung. You can find differences between different populations. there is a mouse microbiome, which I'll show you one slide on, and you can see some sort of relationship between the microbiome and an inflammatory mediator. And I would argue that if you think, if this was all just contamination, you should not be able to find a relationship, for example, between a proteobacter and a cytokine in a lavage. So how does the microbiome, the environment, and health and disease all come together? How is this microbiome important? So the second half of this talk is basically going to show you examples of how the microbiome can be important in health and disease and how the environment can modify this. The greatest amount of work in this subject has been done in the gut. They're far ahead of the lung in this regard. And there's been using comparative metagenomics. Again, this is shotgun sequencing now. This is not just 16S. When you talk about metagenomics, you're talking about shotgun sequencing. So you not only get the bacteria there, but you get all the bacterial genes that are there, the enzymes that they have, and you get the host response. So when you do this type of shotgun sequencing, there's been links between the gut microbiome and weight, the ability to detoxify carcinogens, does this explain susceptibility to cancer, and impact on immunity, which is my area of interest. And there are lots of relationships between the microbiome, for example, and inflammatory bowel diseases. I'll show you on one slide. But back to OB. OB is an OB mouse, and this mouse is fat because it has a defect in the leptin gene, and it eats all the time, compared to in this case, the C57 black mouse. And I called it Heinz for C57, Heinz 57. So that's why I called Heinz an OB. But anyway, interestingly, when you look at these mice, even though this is a genetically engineered mouse, when these investigators in Nature a few years ago looked at the colonization of the GI tract, they found that this OB mouse was lower by 50% in Bacteroides, which is a phyla-level characteristic, and firmicutes were higher. And what this meant, at least in this model, is that the bacteria that were in this mouse were very efficient digesters of things like starches and polysaccharides. So every nutrient they ate, they got it. They got all the calories. And very interestingly, even though this is a genetic mouse, when they took the gut flora from this mouse, stuck it in a germ-free mouse, and the gut flora from this mouse, stuck it in a germ-free mouse, and fed them exactly the same diet, the ones who got the microbiome from OB were much heavier, much more efficient digester of the nutrients that they were getting. This is just a table. Obviously, I don't expect you to read it, but this is just a table showing all the studies that have looked at various bacteria and how they may be protective against inflammatory bowel diseases. A lot of them are lactobacilli, which I think is interesting. Back to the mouth. We know that bacteria break down arginine and urea to ammonia. We know that if your pH is increased in your mouth, you're less likely to have dental caries. And interestingly, the bacteria that is associated with dental caries is Strep mutans. It has the least amount of these enzymes. And so it is a more acidic environment than if you're colonized with these. So the oral microbiome, it probably defines whether we get caries or not. What about the lung, my area of interest? This was a very early study in the New England Journal of Medicine, looking at the relationship between LPS in a mattress, in a pillow mattress, and susceptibility to asthma. And what this showed is that for atopic asthma, and this is important because this is non-atopic asthma, but this is asthma here associated with allergy. In atopic asthma, the more LPS you had in your pillows that you slept on every night, the less likely you were to have asthma. And this sort of relationship did not exist in non-atopic asthma. And the lead author on that paper, I've met several times now since it's part of our microbiome work, Erika von Mutis, who's in Germany, and she has long been one of the leading proponents of this hygiene hypothesis of asthma and has identified in seminal work that exposure to farming environments, and especially to cows, has a protective effect in developing asthma and allergy later in life. And very interestingly, I didn't know this until I actually met her, Mark Holbreich is an allergist in Indiana. And in Indiana, I don't know whether you have any in Kentucky, but in Indiana, we have lots of Amish. And Mark Holbreich is an allergist in Indianapolis and saw the work by Erica and went out and did basically the same thing in the farming communities of Indiana and found that the Amish hardly ever had asthma. And it's probably because of the environment that they grew up in. And this, again, is getting play. So this is the New York Times from last November, basically proposing that we need to be exposing our kids to the outdoor environment, cow sheds, whatever you want to talk, to try to prevent development of allergies and asthma. How might this be so? I mean, why would there be some sort of connection between this? A few basic immunology slides. We all have toll-like receptors on our antigen-presenting cells, on our immune cells. When an antigen, when a bacteria attaches to an antigen-presenting cell or a macrophage, it does so through these toll-like receptors. The receptor for LPS is a TLR4. Once you engage these receptors, these macrophages release all sorts of inflammatory mediators which are responsible for recruiting cells to the area where the bacteria were. And so one could imagine a scenario where you have a lung and there's bacteria in the lung. And if you have TLR4 engagement, you're going to make something called MIG and IP10. And these are chemokines which are known to recruit Th1 type cells into your lung. We should all aspire to be a Th1 phenotype. The Th1 phenotype is protective. You don't get allergy if you have a Th1 phenotype. It's the granulomatous type of phenotype. In contrast, if you have other organisms that are attaching, for example, you might get eotaxin, and this may lead to the recruitment of Th2 type cells into the lung. And the Th2 type cells are your allergy types of phenotype. When you get exposed to something in your lung, you're getting exposed to an antigen. We all get exposed to aspergillus. We all get exposed to pollen. We all get exposed to cat dander. Why don't we all have allergy? It's because it's whatever your background immunologic state is at the time that you get exposed. So if you have a Th1 type phenotype in your lung and you get exposed to a cat, you're not going to make Th2 type of cytokines and recruit eosinophils and develop allergy. You're just going to have some sort of granulomatous response or nothing. In contrast, if you have a Th2 type of background, you get exposed to the same antigen, you're going to have an allergy type of response. So I think the background immunologic environment upon which you get challenged with an antigen is critically important. And in fact, in Erica's studies, all this protective effect against asthma has to be only occurs if you get exposed in the first year of life. Beyond that, I find that actually pretty discouraging because if you're thinking about potential therapeutics and probiotics, it's somewhat discouraging to think that you are already colonized and you can't change how your immune response might be, at least in the lung. And then the latest kids on the block are Tregs. And Tregs are very important in downregulating immune responses, and there's a lot of interest, especially in the gut, how bacteria will induce certain sorts of chemokines and recruit the Treg cells. And just one mouse study, this is stuff from our lab, where we've looked at the bacteria phyla in a bulb C mouse, which is an asthma-sensitive mouse, and a C57 black mouse, which is an asthma-resistant mouse, and look at how diverse the bulb C mouse is. And this has been shown in several studies that the more diverse your background, the more susceptible you are to asthma. But look at the C57. It's dominated by this Proteobacter. Proteobacter is the prototypical LPS organism. It's gram-negative rods. So this mouse has mostly gram-negative rods, lots of LPS. I would argue that this is inducing a Th1 type phenotype in this lung, and then it's harder to get them to have an asthma response, in contrast to the bulb C mouse. We also do some work in Malawi. And this is my fellow who's done some work there. And we've looked at the environment in Malawi and how it leads to differences in the microbiome. So Malawi is the third poorest country in the world. It's in rural South Africa. And we looked at a respiratory microbiome in a BAL fluid in the U.S. as well as the Malawi population. What you can see here, which is interesting, is that the U.S. population is fairly uniform, whereas the beta diversity, or the spreading out of the Malawi population, is very large. The Malawi population has a lot more proteobacter. They have a lot less asthma. Somewhat interesting. This is one of those blots I said you look at and you sort of get glazed eyes when you look at it. But what can you see by a quick scan from the back of the room? You can see if the U.S. population is in green and the Malawi population is in red. There's really two types of Malawi patients, aren't there? There's the ones that look like a U.S. population, and then there's another Malawi population that doesn't have the same thing that we have, but it has this stuff down here. And this actually gets back to this slide a few times ago. There's some Malawians that are like us, but there's some that are way out here. Well, what are these things? This is Petrobacter, and this is Akkermansia. You don't see these things in the U.S. population. Petrobacter was found in 0 out of 20 U.S. patients and in half the Malawi patients. And this was the abundance of the Petrobacter in their lavage. Petrobacter is found in cooking oils. Now, maybe there's some people in Kentucky who still use cooking oils and house fires and kerosenes and stuff like that. Maybe we would find Petrobacter, and maybe in rural Indiana. But at least in this city population, we never see Petrobacter. Akkermansia. Akkermansia was found in 41 out of 44 Malawians and only 1 out of 20. Highly, highly significant. Akkermansia is inversely correlated with weight in rodents and humans. The more Akkermansia you have, the less your body mass index. And in fact, there was an article that came out in PNAS last year that was proposing it could be used in obesity and diabetes because it seemed to reduce the sugar load that people are having. And if you look at the relationship between obesity and the presence of Akkermansia in this population, there's a nice relationship here in the Malawi population. So maybe Akkermansia has something to do with it. Now, I'm not suggesting that Akkermansia in the lung is necessarily causing this, but I will submit to you that the Akkermansia in the lung could also reflect Akkermansia in the gut. I think that we are colonized with similar things. When you eat, some goes down into your stomach, some goes down into your lungs. You colonize, anybody who's done any research in aspiration and pneumonia knows that before you aspirate stuff into your lung, you first colonize the back of your throat. So anything you eat is going to colonize the back of your throat, and then you can aspirate it. So there's probably some link between your gut and your GI tract and your lung. But furthermore, studies are now showing that immune modulation in the gut can lead to changes in the lung, either through immune modulation or bacterial translocation. And basically, this is whole basis for probiotics that we're now looking at these days. So the final part of my talk is going to get back to what I do, and that's HIV. The lung microbiome really started as this lung HIV microbiome project. And this was the first money that was dedicated to studying of the respiratory microbiome. A little aside here, I would encourage anybody who does science, if you ever get a sample from a patient, do what you need with it and then stick the rest of it in the freezer. The reason I got this grant, I am absolutely convinced, is because, as Jesse said, I've been doing HIV research since 1987 as a fellow. Everything I ever got, I stuck in the freezer. And so when this project came out, I was able to go and say, I've got 400 samples ready to be analyzed with good longitudinal follow-up, and the rest is history. That's how I became a microbiome expert. I had no idea what a microbiome is, but I knew I had a lot of HIV samples in my freezer. And when you look at an HIV population, You can see here, in this example, the green are the normal population. The blue are an HIV population who are relatively healthy in terms of preserved CD4 counts. The red are a sicker population with lower CD4 counts. And you can see that these are clearly separate from the others. This is another way to look at this type of data. And I'm just showing you this as a type of data you may see in a manuscript. This is the colors are the same in terms of normal HIV, relatively preserved, HIV sick. What this shows is what are the major bacteria in these populations. So, for example, firmicutes and fusobacteria, the arrows here, are all going towards the normal. Those are what we normally are colonized with. Whereas the sicker HIV population had these sort of purple arrows going towards them. That means they have more proteobacter in their BAL. So this is just a way you may see this type of information. Because we had longitudinal data, we were able to look at patients before 1 month, 1 year, and 3 years after starting antiretroviral, and look what happens. You have this pretty broad beta diversity up front. This person's much different than this, and so on. And with treatment, they seem to be coming closer together. And you actually can do statistics on this, looking at the average distance between each of these dots. And you can see that they become more and more narrow. They less and less beta diversity with time. And you can almost imagine that what's happening as these red dots are being brought back into the fold, so to speak. They're becoming more normal with time on treatment with antiretroviral therapy. You find some unique organisms in HIV. This is one of the first LHMP papers that came out last year, and we're at Indiana. And there's this nice correlation. What we were finding is T. whipuli. And we found it a lot in HIV patients, but not in the normal population. And I was telling this morning, you're allowed 1 blot per grand rounds. That was the rule, I was told. And so here's the blot. This is basically showing a PCR of T. whipuli, just confirming that the sequences in the PCR are correlating here. And at IU, we have patients that are heavily, some of our HIV patients who are heavily colonized with T. whipuli in the lung, shown in yellow here. And interestingly, with time on therapy, it seems to go away. All right, so my final few slides gets to a project that Jesse is actually part of in the latest consortium that we're involved in. And I've talked a lot about bacteria, but it's important that viruses are also part of the microbiome. And there was an RFA that came out that Jesse applied to and got, and I applied to the same group, looking at the late-term complications of HIV in the antiretroviral era. As you know, because of antiretroviral therapy, we don't get opportunistic infections anymore. What you're seeing are inflammatory type of infections. So in the lung, we see COPD, pulmonary hypertension, neoplasms, and infections are much less. And then you have all these vascular complications. And these are the things that you see in the heart era. It's not so much infections. So why is that? Well, our hypothesis in our project is that this is all immunosenescence, accelerated aging, that in HIV population, if you look at their T cells, they're all immunosenescent. They're old. They've replicated many times. And an immunosenescent cell secretes an inflammatory type of mediator profile, a lot of IL-1, a lot of TNF. These are the cytokines that induce this generalized inflammation that lead to type of vascular disease. What drives this immunosenescence? Anybody ever had a latent virus? Viruses drive latent, these sort of chronic antigenic stimulation. Bacteria you can treat and they go away, except for some like mycobacteria, some of your interests. But viruses, they tend to hang around. And as long as they're latent, as long as they have proteins that can be expressed, they can sort of chronically stimulate the immune system, leading to this chronic inflammation, these chronic long-term complications. So we hypothesized that either HIV or other viruses in these patients persist despite antiretroviral therapy, and they're leading to chronic inflammation, which leads to these long-term complications. And we have some very preliminary data showing that in these 3 HIV-positive patients, who are, at least these 2 are relatively well controlled, had these lymphocryptovirus. This is EBV. So EBV was present in 2 out of the 3 HIV-positive and really none of the normals. So you do have this latent virus in this population despite viral control of their viral loads. And interestingly, when you look at these positive patients who have this EBV, they have the highest mediator concentrations, whether you're looking at IP10 or MCP1. They have these chronic cytokines that are chronically inducing inflammation, that have the potential to induce chronic inflammation. And so our project in this new consortium, of which Jesse's a part of, our focus will be looking at for the existence of persistent virus in people on antiretroviral therapy and relating it to the complications we are detecting on CT scanning, pulmonary function, and questionnaires. So I'm going to end at this point. I would like to close by saying that we are in an environment where we have to live happily with the bacteria we are colonized with. And the challenge is there ways we can manipulate our microbiome to make us better, to make us more efficient at metabolism or make us less susceptible to certain diseases? But at the same time, we have to be aware that some bacteria that we are colonized with and some bacteria that we can administer have potential to make us sick. So there's this obvious balance that is necessary, but the microbiome, I believe, is here to stay, and it is an important part of defining who we are in health and disease. And with that, I would again like to thank Jesse for inviting me, and I'd be remiss if I didn't thank all the people that are associated with my work in HIV and with the people in Africa. So, I'd be happy to take questions.
Thank you, Homer. So there were a couple of concepts that you brought up that I want to emphasize. One is the fact that you collected in the freezer everything you did. And he mentioned that I'm involved in this project as well. It turns out this is all the ID group with Julio Ramirez and his team. They have the largest HIV program in Kentucky, as you know, with over 1,800 patients. And they collect data all the time. So when we applied, Julio and I sat in a room and just talked about, let's put this all together. And so we're part of this consortium. It allowed us to begin to learn about things about like the microbiome and oxygen stress and other things. And this will be quite exciting. So I thank the ID group for actually doing what you do. The second thing that I wanted to bring up as a question is this concept of bacteria changing the way our bodies and our cells behave. And it brought this concept of epigenomics to me. Because epigenomics, as you know, is alterations in the expression of genes that has nothing to do with the sequence of that gene. No, it's about something else. And so I wonder if this concept of the microbiome is going to be touching the concept of epigenomics, because the microbiome actually may be altering how some of these genes express. Can you tell me about that?
Well, I know people are looking at that. They're looking at methylation of genes and regulation of genes by other bacterial products. I'm sure it's related. I don't have any data yet in this nascent field. It wouldn't surprise me, though, if they weren't related. There's another study going on at IU that's sort of along those lines, and it's in the GU track. And what they're studying is whether you get certain sexually transmitted diseases. And what they found is that if you get a certain, I believe they're looking at chlamydia, but everybody's getting exposed to chlamydia. Why do just some people get it? And what they're finding is that it's what you're colonized in the first place with, the non-important bacteria that create this environment that allows the chlamydia genes to be expressed. And some of it has to do with the pH of the environment and whether that's allowing the genes in the chlamydia species to be expressed or not. But that may sort of in a roundabout way address some of what you're saying, but the control of gene expression through epigenetics is obviously highly important. But
Questions? I don't see hands. Questions? Yes, sir.
You emphasize that there are differences in pathological states between specific bacteria.
Yeah.
So singular bacteria can do something and that might be harmful. But is there evidence that there is actually interaction between different bacterial species within the same host?
oh, sure.
The same bacteria would be harmful in one context and not on the other. Doesn't entirely depend upon one particular unique bacterial species being there, but a whole difference in ecosystem.
Well, that's what Clostridia is, almost. What you do is, we're all colonized with Clostridia. What happens, though, is the other bacteria suppress it. You get rid of the other bacteria, it's now pathogenic. Now, does Clostridia by itself convey any sort of benefit to us? I'm not a GI microbiome person. If there's any GI doctor, I'd be curious to see what they say, but my suspicion is it probably does. There's probably some digestive enzymes that are part of Clostridia. It has some role, but it does become pathogenic when it becomes predominant. And I'm going to argue in the lung that that's what pneumonia is. I think Proteobacter absolutely essential. I want to be colonized with Proteobacter because I'm going to have a Th1 type response. But Proteobacter are gram-negative rods, so obviously patients who have cystic fibrosis, there's lots of Pseudomonas around. It's one of the slides. I didn't go into detail. So it's a matter of proportion and the other bacteria keeping them under control.
We have some GI people here. Dr. McLean.
So there's a lot of difference between the lung and the GI tract.
Yeah.
So the things that you like that are good in the lung are actually bad in the GI tract. So you get an Proteobacter, that's bad. And take a possibility of generate alcohol. So you can have a lot of organ that's the microbiome. And I think that if you how do you get the right bacteria or virus growing in the right organ?
You're absolutely correct. It was interesting to watch the evolution of this field. 3, maybe 4 years ago at the ATS, they had a lung microbiome symposium at the American Thoracic Society. 10 speakers, 9 of them got up and talked about the gut. And they all said, well, it's by what's going on in the gut is inducing immune regulation in the lung, either through creating T regulatory type cells or inflammatory mediators. Only one person came up and even talked about the lung. The last 2 years at the lung microbiome session, it's been mostly lung. But it is interesting that the gut it's completely different in terms of what you're colonized with. Though you really don't often find stuff in the lung that's not also in the gut. It's just in the relative proportions.
Tell me about this concept about perinatal origins, because there's a lot of interest in perinatal origins of asthma. And this concept that I think you said you got impressed with that, and I did too, about the fact that after the first or second year of life, you're done.
You're done.
No matter what you do, it's sort of. So are we going to be able to manipulate these things in a way that have a significant impact on disease?
Ask me after my grant submission. Because really what we're doing, I showed the mouse model. What we're trying to do is exactly that. And I didn't show data because this was general audience, but we can get rid of the microbiome completely by using nebulized tobramycin. And the next goal is to reconstitute that environment and see if you change susceptibility to antigens. My speculation is that you're going to have to do this in a very young mouse to do that. Because that if you do it in an adult mouse, it's too late. That's my working hypothesis after listening to Erica talk, that we're not going to be able to change it in adults, which is discouraging.
Yeah, absolutely. Final question? have something here for you. We have an official thing and something more fun. So this is a plaque, okay? We want to give you in honor of the named lecture. It's the 18th annual Nancy Middleton Smith Lecture, presented March 6th to Homer Twigg. And the fun part is the back. You've heard perhaps that we are committed to having every single institution in the United States having someone with a slugger bat from Louisville. And it says, no, you can't touch it. Twigg, 2014 NM Smith Lecture, University of Louisville. Thank you so much.
Thank you.
Thank you, everybody. Have a good day.
That's nice. Yeah.
Very wide.